Today we'll talk about this paper that studies the role of these MMP, that stands for matrix metalloprotease. So MMPs are a fairly wide family of proteases that typically exist outside of cells. And this particular one, MT5 MMP, it turns out that it goes by an AKA, Sp26 I think it is, but the rest of the paper refers to it as MT5, so that's what I'll do as we discuss the results today. So it's a paper on MT5 and Alzheimer's disease, and the title of the paper is that MT5 promotes neuroinflammation, neuronal excitability, and amyloid beta production in primary neuron astrocyte cultures from these mice that are prone to developing Alzheimer's disease because they have a series of transgenes in them that will make them more prone to developing Alzheimer's, right? As we'll see later today, I'm not 100% in agreement with the title of the paper. I think the title of the paper is being a little bit more aggressive than what the data actually show, but that's the title. So in the introduction, the authors mentioned that MT5 MMP, aka MMP24, is the only MMP that is preferentially expressed in the nervous system. So whenever you see something that is expressed preferentially in an organ system or a cell type, there is this intuition that it must be doing something important there, right? If it were doing something really important everywhere, it would be expressed everywhere. And if it weren't doing anything really important in this particular organ or tissue, why would the organism bother expressing it preferentially here, right? So there's this intuition that it doesn't always have to be true, but that normally pans out, that whenever there is a gene or a protein that is preferentially expressed in a system, there is the likelihood that it's doing something critical. The authors in the introduction also mentioned that only a handful of its substrates have been identified. These MMPs typically have a wide range of substrates, so that they exist outside of cells, normally chewing up on a number of things. And to identify those things, generally it takes like some sort of careful screening for potential substrates. And for MT5, only a handful have been identified so far. And perhaps the most important thing is that APP is one of them, right? So APP is a demonstrated substrate of MT5, and MT5 deficiency markedly reduced the accumulation of amyloid beta peptides in these AD-prone mice. That was an observation that had been made prior to this paper, right? And it is a pretty important observation because we're trying to understand how MT5 may contribute to one way or another to the development of Alzheimer's disease. And we have this previous observation saying that mice that lack MT5 produce less amyloid beta. So right off the bat, they know that MT5 may have a very important role in developing the disease. Now, here's the statement or the observation that connects with the rest of the paper, if you will, and that is that MT5 deficiency also prevented increased levels of IL-1 beta, interleukin-1 beta, in glia of these mice. So if you guys remember, AD or Alzheimer's is about these amyloid beta plaques and the development of neurofibrillary tangles. Neurofibrillary tangles made by phosphorylated tau. But that's sort of the end point, right? One of the main drivers leading to the accumulation of these toxic protein or peptide complexes are neuroinflammation and neuronal hyperexcitability, right? Which may go hand in hand. So the idea here is that yes, what breaks neurons in the brain are these neurofibrillary tangles and the amyloid beta plaques. What leads to the accumulation of them is neuroinflammation. So seeing that MT5 deficiency prevented the increased levels of interleukin-1 beta and glia of these mice that are prone to developing the disease, somehow suggested that MT5 may also have something to do with neuroinflammation. So, not necessarily a leap of faith here, but the authors hypothesized that MT5 may operate at various levels of Alzheimer's disease progression by regulating APP metabolism in a way they saw that already, IL-1 beta expression or function, and neuronal function as well. So that's sort of the third missing leg in all of this. Questions so far? No. So the sort of the gist of the rationale behind this paper is we've made these observations linking MT5 to amyloid beta accumulation in the brain of these mice, linking MT5 to potentially neuroinflammation in the brain of these mice. We're going to actually more actively put forth the hypothesis that MT5 will operate at all three levels of Alzheimer's disease progression: neuroinflammation, amyloidogenesis, meaning the formation of these amyloid beta peptides and plaques, and neuronal function or hyperexcitability. Okay? Cool. So first things first, they take a look at how, because like if you recall some of these preliminary observations they made in mice that are prone to developing AD. So they first want to see how the progression towards AD may affect the expression of MT5. Before they get into how MT5 may affect AD progression, they look at how AD progression may affect MT5. So they look at the mRNA levels of MMP24. It's the only time that they name this gene by its alias. So they're looking at the levels of MMP24 mRNA, and they see that there is no significant difference between wild-type mice or transgenic mice. And anytime that in this paper we see transgenic, what the authors mean is the series of transgenes that will make these mice prone to developing AD. These mice have inserted in their genome a series of APP variants that bear mutations that will make them more prone to be cleaved in a manner that gives the amyloid beta, the toxic versions of amyloid beta, right? So they're transgenic in that they carry APP genes that will more often than not generate toxic amyloid beta peptides. So what you see here is mRNA for MMP24 data for both the wild-type mouse and the mouse carrying these transgenes, making them more prone to developing AD. And what they show us, the black bars correspond to control animals, meaning animals that have no further treatment to them other than those underlying genetic differences. And then in the gray bars, we have, these are, I should take a step back. These are primary cultures of neurons extracted from these mice, right? So these are not the actual whole mice that they're dealing with. These are neurons that they've extracted from these mice and placed in primary cultures. And when they subject those or incubate those neurons, primary culture neurons, with interleukin-1 beta, the main pro-inflammatory cytokine in the brain, they see that there are no significant changes within either genotype in the mRNA levels of MMP24. They also look at the protein levels. So these MT5-OD means optical density, and this is the classic densitometry based on a Western blot. So I'm not showing the Western blots here, they're shown in the paper, but essentially what they did is they did a Western blot for MT5 protein, and then they measure via densitometry the relative amounts of the protein present in each of these samples. And here what you can see is that the transgenic animals have more protein overall than the wild-type neurons. And again, there was no difference whether the cells were exposed to interleukin-1 beta or not. So, why is it not showing up? There you go. So the, oh, I know it's not showing up. It's not that it's not showing up. It's that this thing is a little too high. There we go. So the conclusion from these two experiments would be that there is no difference in MT5 mRNA between wild-type and transgenic cells, but there is more MT5 protein in the transgenic cells than in wild types, right? And that MT5 is not induced by interleukin-1 beta treatment and therefore may not respond to neuroinflammation. My question to you guys is, how can the protein levels in the transgenics be significantly higher than in wild types if they both have the same amount of mRNA? So translation efficiency, all right. Hold on one second, because I had a...
There could be like a regulator in the protein levels in response to the transgenic mice.
Uh-huh.
It could be something that's avoiding it or like preventing its degradation.
Preventing its degradation, so less degradation. We're gonna present these from the perspective of the transgenics, right? So either the transgenic cells may be better at translating them, or they may be they may be worse at degrading them. You said, yeah? Post-transcriptional what?
That's all I got.
That's all you got. The issue with post-transcriptional regulation, this idea that, well, maybe they're transcribing the gene at the same rate, but the transgenic cells are somehow better at keeping that mRNA or making it more stable, or all of that would show up in the measurements of mRNA. If the transgenic cells and the wild-type cells were transcribing the gene at the same rate, but somehow the transgenic cells were better at keeping that mRNA around, when you extract the mRNA from these samples, you should see significantly more mRNA in the transgenic cells.
So, you mean the transgenic mice, they are more AD prone, right?
Mm-hmm.
So, there's a mutation in them, like for AD, like genes that were introduced into them?
Yeah, so we've seen these mice before because they're pretty common in Alzheimer's research. The 5X FAD mice, I forget the exact details, but essentially these mice are transgenic in that they carry a bunch of genes that normally mice don't have, right? So someone introduced into them these transgenes, and these transgenes are variants of the APP gene with mutations that make those APP proteins more prone to be cleaved in a manner that generates the toxic forms of A beta. Right? So they're more prone to developing the disease the same way that humans with mutations associated with familial AD are, in that they carry mutations that will make those APP proteins more prone to be cleaved the quote-unquote wrong way or the toxic way. Okay? So the idea behind these transgenic mice is the mutation will cause accumulation of toxic A beta, right? And then that accumulation of toxic A beta would probably kickstart the whole domino cascade of events that we've been seeing over and over throughout the semester, right? So more A beta means that the microglial cells might detect those increases in A beta and respond with neuroinflammation. More A beta also may mean, through a series of mechanisms, that more calcium gets into the neurons, making them hyperexcitable, right? So now you have these two legs of the problem being triggered by the same initial accumulation of A beta. But let's talk about inducers. So when you said inducers, I thought that you were going to go either of two ways. One, inducer of transcription, which we already touched on, so it can't be because it would show up in the mRNA levels, or an inducer of MT5's activity. Exactly. So looking at the protein levels in the transgenic are higher, but that says nothing about the activity of MT5. When I use a super big word to describe what I think you're describing, extractability, right? So maybe the transgenic makes MT5 more accessible to us as researchers to extract it from the samples. And that may sound like, wait, hold on a second. I thought that we were looking at protein, period. Like, what does it mean to be more extractable? Well, the reality of it is that there are many proteins. I don't think MT5 is one of them, though. But there are many proteins that will form super high molecular weight macrocomplexes. And sometimes some of the techniques that we normally use to isolate proteins from cells won't work in them, right? So it's a technical difficulty, but I don't see why not. It might not be affected by the environment within the transgenic cells. Okay, so a couple of things. So inhibitors of translation in general?
Or
Specifically of MT5. Everybody's stealing from you, Jessica. This is so unfair. It's all flavors of the same thing, that somehow in the transgenic cells, the same mRNAs are being translated more efficiently, or the protein is being degraded less. Or the reason I asked what kind of translation disinhibition you were referring to is, let's say that the transgenics are better at translating, period. If that were the case, then you would have more MT5 in the transgenic cells, but you would have more of everything, right? And if you had more of everything, that would include housekeeping genes. And if you guys remember, these densitometry analyses are always made relative to housekeeping genes like actin or GAPDH or tubulin, right? So if there is more MT5, but also more of any of the housekeeping genes that we normally use to make levels of proteins relative to something, to some sort of reference, then those bars would be just as high as in the wild type. Because in the wild type, you would have less MT5, but also less actin. In the transgenic, you would have more MT5, more actin. The ratio between the two may be the same. More efficient translation. I've heard that before. Now, look, guys, we've pretty much exhausted about everything there is, right? So whenever you see increased levels of a protein, it can be that can be because it's being produced more, so higher translation efficiency, because it's being degraded less, or this interesting idea about how accessible it is to us as human beings trying to get these things out of the cells. That's about it. And of course, these are symmetric in the sense that it could be higher translation efficiency in the transgenics or worse translation efficiency in the controls. The less degradation in the transgenics could be that this protein is more effectively targeted for degradation in the controls. Either way, the outcome will be the same, and that is that there's more of this protein in the transgenics than in the controls. Yeah? All right, cool. So, let me, oops. Done, whatever. Just want to get your names down. Wells, Andre. And here I have, if I forgot you, let me know later. Jessica. Of all people, the universe is conspiring against you today, Jessica, so. But here's where I wanted to get into, because here's where the data start getting really interesting. So, then the researchers turned the question around, and instead of asking what does AD progression do to MT5 expression or levels, they asked the question, what does MT5 do to AD progression, right? And if you remember, they're interested in these three angles, right? The neuroinflammation angle, the neuron excitability angle and the APP metabolism angle, the production of amyloid beta peptides. So they start with neuroinflammation and they ask, how does the presence of MT5 affect the expression of these pro-inflammatory cytokines that are produced by cells and that can drive AD progression when they're produced in excess, right? So they take a look at IL-1 beta mRNA in wild types that are missing the MT5 gene. So these are MT5 knockout mice from which they extract these neurons. Then they take a look at the neurons from transgenic mice, these AD-prone mice, and then AD-prone mice that miss the MT5 gene, right? And what they see is that it seems like when you look at the wild types, what they point out is that there is no significant difference in the amount of interleukin-1 beta mRNA produced between these two cell or genotypes. So taking out MT5 does not affect the levels of interleukin-1 beta mRNA levels. But when you look at what happens to the IL-1 beta mRNA in the context of these transgenic mice, the lack of MT5 significantly reduces the amount of IL-1 beta, at least mRNA, being produced, right? So there's two things that I'd like to discuss with you now. One is, how can MT5 be required for IL-1B expression in the transgenics but not in controls? That's what these data seem to indicate, that you take out MT5 in controls, nothing happens, so it's not required, but you take it out in transgenics and now all of a sudden you lose IL-1 beta expression. So how can that happen? Yeah.
Okay. Do you mean both on like WT and TG? Like, are you asking specifically for that?
The comparison between the two. In WT, in wild types, you take out MT5 and nothing happens to IL-1 beta expression.
I think it's because is it the 1L? How do you say that? The expression?
IL-1, yeah.
The WT expression, like it's normal.
Uh-huh.
And I think the TG is high, so if you remove it, it's high, so it's causing higher levels. And
Uh-huh.
if you remove the MT5,
5.
drop it.
Okay.
It needs a pathway for that. So they're dealing with stress, or there's something there that WT doesn't have that's causing it to spike.
Okay. Did everyone follow this argument? The idea here is in wild types, MT5 is not required for IL-1 expression. In transgenics, there is an increase in expression. So in the transgenic, there must be additional things that are happening that contribute to IL-1 expression. And for those additional things, MT5 is required. Right? So the idea here is that in a wild type, I'm going to try and paraphrase this to the best of my ability. So in a wild type, you have IL-1 beta expression, and MT5 is not contributing to it by itself. There's no room for MT5's contributions to expressing IL-1 beta. But something in the transgenic makes that MT5 is now needed for that induction in IL-1 beta expression. Right? Now MT5 is somewhere in the pathway connecting the transgenes in these mice to the increased expression in IL-1 beta.
In the last column though, when MT5 is present, the levels go, or is it present, the levels go down?
When you say the last,
The last bar.
yeah, this one here,
Yeah. So
yeah.
wouldn't that mean that? Oh, okay, never mind. No, I got it.
Yeah, yeah. So based on Lara's hypothesis, in this transgenic that lack MT5, there is now these cells that all of a sudden needed MT5 for TG to connect to increase IL-1 beta expression. They're missing that intermediate, so that connection is lost. Okay? Yeah, what's that?
If that were true, wouldn't level is the wild type MPs?
So what you are suggesting is that these two, let me see if I can, that those two should be the same,
Right.
right? Beautiful, excellent. Excellent observation. If Lara's model is correct, then when you take out that intermediate between TG and IL-1 beta expression, what you're doing is you're negating the whatever the transgenic added or TG added to expression of IL-1 beta. So you should bring it back to the levels in controls, not much lower. What do you think? Is this significantly lower than that? I see some heads shaking yes, right? So the question is this, to use their notation, sorry about that. To use their notation, is this also statistically significant? Why do you think that is the case, Jacob?
It's the deviation of 0.5 and above one right now seems statistically different because, in my opinion, the first order.
Anyone else for, yeah, they seem statistically significantly different to me. Yeah? Why?
I mean, it seems like a big gap, but I think with the error bars, if it's not statistically significant, they should overlap.
Overlap, right? How many of us heard that argument that if the error bars don't overlap, then the differences between the groups are statistically significant? Gabi, you're like, no. Well, it turns out you're right, so say something. Very, let me see, it's in very unique situations that the lack of overlap between error bars indicates statistical significance. It all comes down to what those error bars are depicting. So if they're depicting, let's say, your 95% confidence interval, then sure. But if they're depicting standard deviation, for instance, or in some cases standard error of the mean, that may not apply. And I think, Jacob, I think that you started hinting at that. When we compare those two, and this gets to the heart of statistical comparisons when you have more than two groups, right? Here we have 4 experimental groups, right? You might say, okay, well, that's significantly lower than that, but you're not taking into account variability across all groups, right? When you look at those two by themselves, you might say, wow, that bar is a lot lower than the control. But then there is the second bar that is a little lower and the third bar that is a little higher, right? So statistical tests will take into account, proper statistical testing will take into account this variability across groups. And in that context is that it may find that, yeah, you know what? I see so much variation across all groups. I see bars going up and down so much that the only two that I feel confident there is a real statistically significant difference are TG and the TG mutant MT5. Every other comparison, there's so much noise in the data here that every other comparison, I don't think they may be different, but I just don't have the confidence to say that they are. Does that make sense? So that's why they don't pass a statistical significance test. Okay? So back to whether or not these would be statistically significant or not. If they were, they would have shown us. There would have been a little bracket linking those two and indicating that they were statistically different. So the fact that they don't says that they probably ran the test and there was no difference. So far, so good? The other thing that I wanted to discuss with you is the concept of, so the way that the authors analyze these data, they tell us in the paper, is by something called ANOVA. You may have heard of this term before. If you haven't, no problem. ANOVA is a fairly common statistical test by which you compare different data sets from different groups based on the overall variance within each of those groups and across all samples. That's why it's called ANOVA, which stands for Analysis of Variance. Okay? So when you do ANOVA, essentially you're doing what I just described. You're saying, all right, I'm going to look at all the data sets. I'm going to look at variability among them. And based on that overall degree of variability, I'm going to assess how significant any pairwise differences may be. If there's a lot of variation across all groups, any two groups that I choose to compare pairwise may be different from one another, but that difference may not mean much given all the variation there is in the background. If, on the other hand, every group is about the same as everybody else except for one bar that is, it doesn't even have to be super low. It may be just slightly lower. That may give you a statistical significance for those comparisons. So that's what we refer to very generally to as ANOVA. Yeah?
Since they use ANOVA, was the variable whether or not they had MT5, or was it whether or not?
Oh, excellent question. Excellent question. So the question here is, if they're using ANOVA, what was the variable? And the answer to that question, so Jacob is asking, was the variable whether they had MT5 or not, or was the variable whether they were transgenic or not, right? The answer to the question is, if you use ANOVA, the variable is genetic background. And it's a variable that can take in on 4 different flavors in this particular experiment. Genetic background can be wild type, or MT5 minus, or transgenic, or both. But a simple ANOVA would treat each of these as independent from one another. Your intuition, though, is, well, but hold on a second, they're not independent. I mean, in this guy here, I have both the 2 other manipulations, right? So they're not like totally independent from one another, correct? You're absolutely correct. And that's why we have something called two-way ANOVA or two-factor ANOVA. Now, if by show of hands, has anyone heard of two-factor ANOVA before? Helen, was that a timid yes? You heard of it? Would you be able to explain it? Exactly, that's exactly what it is. It's a two-variable ANOVA. In a way, going back to what you were asking before. So it's an ANOVA that says, instead of treating each group as different genotype and making the one variable genotype, what I'll do is I'll classify these groups along 2 variables. So whether or not they have MT5 and whether or not they're transgenic. So wild type, for instance, is a no for lacking MT5 and is a no for transgenic. MT5 minus minus is a yes for lacking MT5, but it's a no for transgenic. The transgenic is a no for lacking MT5, but a yes for transgenic, and the transgenic MT5 knockout is a yes for both. So what the test will do is it will say, all right, let me first look at the effect of lacking MT5. So it will group, mathematically, all of this is done mathematically, it will group the data from both MT5 minus samples, whether in a wild type or a transgenic environment, and compare them to the 2 that do have MT5. And it will ask the question, is the difference here? Taking out MT5 globally, does it cause a drop in the expression of IL-1 beta? Then it will do the same for the other variable. So it will group together the 2 that don't have the transgenes and the 2 that do have the transgenes and answer the question, having the transgenes, what does that do to IL-1 beta expression? And then finally, most importantly, it will do an interaction between these 2 factors or variables. So what it does is it says, based on what I learn about my MT5 level comparison and what I learn about my transgenic level comparison, what should I expect when I combine both? And then it mathematically compares the observed results with the expected results, and as usual, with any other statistical test, it will determine what are the chances, what is the probability that the differences that you see are due to chance or real. Okay? So in this case, I don't know about you guys, but I think that if we were to pull these, the second and fourth bars together, overall, those are lower than the first and the third. So I don't know, I can't really speak for a statistical test that we didn't do. But there might be an argument for saying that the lack of MT5 in both leads to less IL-1 beta production, that having the transgenic or not probably makes no difference, because when you look at both no transgenic versus both with the transgenes, the averages between the two may be kind of similar. So transgenes don't do much, so they don't affect IL-1 beta. And if that is true, that transgenes don't do much, and MT5 loss will lead to a drop in IL-1 beta, then the question is by how much do you expect that drop, right? And what a two-way ANOVA might say is, look, I know that losing MT5 causes a slight drop in IL-1 beta, but the drop that I'm seeing here is way more than I expected, right? Based on my other calculations, I expected this bar to be somewhere here, a slight drop, not such a huge drop. So I think these two things are interacting. I think that when you take out MT5 from any generic mouse, you will lose some IL-1 beta expression, but when you take it out from a transgenic, that drop is gigantic. Okay? Did that kind of make sense? So it is very common, we've seen it over and over, it is very common in research to say, I'm going to try variable 1, variable 2, and then variable 1 and 2 together. Anytime that you deal with a situation like this, theoretically, you should use two-way ANOVA, not one-way ANOVA. And yet, you see in literature over and over and over, people using regular one-way ANOVA. Okay? So if you ever want to impress your future PhD supervisors or bosses or, you know, the C-suite in your company, shouldn't we use two-way ANOVA here? And you're going to make some frenemies. They'll hate you for pointing out something that is true, and they'll love you for being on top of things. Okay. Let's move on because we're lagging a little behind. So then they repeated the experiment, but this time they said, fine, but what if we look at the levels of IL-1 beta when IL-1 beta is added to the culture media? In other words, what would happen when we are, in a way, stimulating the neurons in a pro-inflammatory environment? We're telling these neurons, there is inflammation here. How much interleukin-1 beta will they produce then, right? So they repeated the experiment. If you notice, the black bars are pretty much identical to what we were seeing before, except at a slightly different scale. And then they added interleukin-1 beta to the cultures, and what they see is in the wild types, the expression of IL-1 beta shoots up, right? In the transgenics as well. But in the wild type without MT5, there was no expression of IL-1 beta at all, induced by the pro-inflammatory environment. And in the transgenic without MT5 gene, there was some increase, but it wasn't as much as you see in the transgenics with MT5 in them. So my question to you is, what would a two-way ANOVA show here? And let's, for a second here, let's focus on the gray bars. Let's not worry about the black bars. Let's focus on what would happen in cells that are within a pro-inflammatory environment. So what do you think? Will the MT5 show an effect? And again, we're kind of role-playing here. We're pretending to be a statistical analysis package, but what you want to do is pull together the data from both MT5 --, compare them to the data from both MT5 ++. So would MT5 have an effect? What do you think? Gavi, I see like a head nodding yes. Yeah? Why? So in a way, if we were to pull these two together, it would be something like this, whereas if we pull these two together, it was something much higher, right? Like if we were to pull the data between those two groups. And I totally agree. I think that there would be an MT5 effect. What about transgenic? I see a yes here and a no. You guys want to fight over that?
I have a question.
Yeah.
When you say maxed out on that one, would that have anything to do with analyzing whether or not MT5 has an effect? Would that be like the max capacity that they can see?
Where do you see they're maxing out?
So the IO on the IO bars,
Uh-huh.
they both have the same. Would that be like the maxing out at a specific level? Or?
So I think it may look to you the same. Like if you get a little closer, this one is a little higher. It could be that they're approaching either maximum detection or maximum production by the cells. It may be that these poor cells, there's so much mRNA that they can produce, right? But when you get closer to the graph, they're not like the exact same. So what do you think about transgenic? Will transgenic affect the expression of IL-1 beta in the presence of IL-1 beta? No? Why do you think it won't?
There's not enough deviation between the rate bars.
Enough deviation or enough more like difference, right? So to look at the potential effect of the transgenic, what we do is we pull these two together, and that's going to be somewhere in there. And then we pull these two together, and that's going to be somewhere in there. So I suspect that there wouldn't be much of an effect, right? Would there be an interaction, though? So the question here is, so what you're saying is transgenics would rely more on MT5 to produce IL-1 beta in response to IL-1 beta, right? Yet what these data are showing is that if in a non-transgenic, when you take out MT5, you completely kill the ability of these cells to produce more IL-1 beta. They produce just as much as in cells that are not exposed to IL-1 beta. Here, they still produce more, right? So what that would mean is that in the transgenics, they rely less on MT5 to produce IL-1 beta. Does that make sense? Yeah? I can see how it's getting a little overwhelming. So let's take a step back and simply say this. One of the things that both this experiment and the previous one highlight or emphasize is the fact that you get all of these interactions that we're finding. So I think that MT5 has an effect, transgenic doesn't. So when you look at the double, right, you would expect that MT5 has an effect. Transgenic doesn't do anything. MT5 drops the production of IL-1. So you would expect that in the double, the production of IL-1 drops compared to the transgenic alone. And it does. But does it drop as much as you expected or not? We would have to run the statistical test. The point here is this, that just by looking at some of these bars, you can see how the behavior changes depending on what genetic background you're in. And that is something that the authors figure out rather quickly, that it's not as simple as to ask, what does MT5 do to these cells? Because the answer varies a lot depending on whether you're in a wild-type environment or a transgenic environment. When it comes to this particular experiment or this particular biological readout, the absence of MT5 was absolutely lethal, right? It completely killed the ability of neurons to respond to a pro-inflammatory environment by producing more IL-1, right? Those neurons were done. But in a transgenic environment, not so much. We can link that to what Lara was pointing out before. In a transgenic environment, there is this increased production of IL-1 that it depends on MT5. And this is the baseline, right? So this the black bar here. that depends on MT5, even though in the wild-type environment, the production of IL-1, baseline IL-1 production, did not depend on MT5 at all. So both of these observations speak to what I like to call, and I think, yeah, I have it here in the title, a rewiring of the gene regulatory network at play in these transgenic mice. There is some sort of rewiring of the network of genetic connections that will, for instance, connect the transgenes to IL-1 production in baseline conditions, the IL-1 production in response to pro-inflammatory signals, and its dependency on MT5 or not, because for baseline, IL-1 production was depending on MT5 in the transgenics, but for induced, it wasn't. Right? So there's a complete rewiring of the network and the logic behind all of these regulatory things. Does that make sense?
So say, can someone be more susceptible from like outside factors?
Depends of quote unquote when you ask, right? So what this would mean is that if you were to inhibit MT5, let's say that you decide that inhibiting MT5 is good because it reduces neuroinflammation. So you'd like to form a company trying to sell a drug that will inhibit IL-1 beta or neuroinflammation based on inhibiting MT5. Then cool. Whoever read this paper will say, well, hold on, because inhibiting MT5 will reduce the levels of IL-1 production under baseline conditions. But if you have early Alzheimer's disease, if you already have some of the features that these transgenic mice will have, right? So that sounds great, right? That sounds awesome. I'm giving a drug that will only work in people who may be experiencing early signs of the disease. Sounds amazing. It turns out that those same brains, the second that they start getting a little inflamed, they're not going to respond to the drug anymore because you're inhibiting MT5 here and it's not really doing much to IL-1B production. I mean, it is when you compare it to with MT5, but not as much as what you achieve by inhibiting it in a controlled situation. So altogether, it may be one of those super complex situations in which, yeah, you will give the drug, but the outcome that you get is very dependent on a whole bunch of previous conditions. Yeah? That makes sense? Okay. Maybe we have time for one more or two. Well, here, what I meant to do here is sort of put it all in one graphical representation. If we split the world of mouse brains between wild type and transgenic, AD prone, it seems like in the wild type brain, MT5 normally does not contribute to IL-1 beta production, right? It's just not doing much in the absence of neuroinflammation. When we take out MT5, nothing happens, not much happens to the production of IL-1. Those are the black bars. But when we add IL-1 beta, it seems like that induction of IL-1 beta by IL-1 is dependent on MT5. Because if you take out MT5, like we did here, now that induction is gone. Or rather the induction. The expression is still there, but the induction is gone. Okay? In a transgenic world, MT5 is normally connected to IL-1 beta expression, perhaps because the presence of those transgenes kick-started a whole bunch of new pathways that depended on MT5. So MT5 is connected to IL-1 beta production, hence that significant drop that we see in the absence of inflammation. But when you add IL-1 beta, there is an increase that may be similar to the increase that you see in the presence of MT5. And that is telling you that whether MT5 is there or not, the increase of IL-1 beta expression induced by IL-1 itself is independent of MT5. It's going through some other mechanism. And look at what we did here. There is some wiring and a different wiring. We've rewired the network.
Maybe a positive feedback loop if you added that then.
Absolutely. So you're pointing to this, right? Because the more ILB1 that is expressed, the more that it will induce more ILB1 expression. Yeah, it's here and it's here. So in both cases, whether MT5 is there or not, the presence of this cytokine triggers a self-amplifying autoregulatory loop. Yeah.